lockout. Please, sir.
Okay, so next week we will have some off because of the holiday, Muslim holiday. So this one is before we take a break. We will continue to reproduce pipeline, and also we will continue to learn by Python. So it's very important for you, this library, because it's widely used to access the NCBI and perform some basic analysis for sequencing data. Okay, so let me start maybe 15 minutes and I will hand over to Fadin and Sinta to continue with pipeline. I think it's still on Galaxy or maybe already moved to other framework or platform. So our roadmap is to be able to reproduce Galaxy and also reproduce the snake make. It's a Python disk, and then Nextflow. Okay. So before, maybe you already tried this library, just pip install Biopython, and you can just quick start sequence object, sequence annotation, sequence input and output, how you read the data from public data bank, and then perform some input and output. You can also do alignment. Alignment is how you can align your, assuming the sequence becoming a string, you can align, creating alignment object for sequence and coordinates, and slicing, indexing is like a string operation in computer science, but this is more specific for sequencing data, and you can also do some operation and alignment. Okay. You can also do more complex alignment, which is pairwise sequence alignment. This one, I will give it to you to try all of this alignment. Make sure that you try the code copy paste at the same time you build your prompt engineering capability. Like once you copy paste, you try to prompt to ChatGPT to make sure and to see whether ChatGPT or Gemini can reproduce your code. Okay. So today I will spend maybe 10 minutes to discuss about BLAST and other sequences tools. It's a very important tools called BLAST in Python. So what is BLAST? So if you want to do comparison between your sequence and other sequence, let's say other sequence already analyzed by some other people, and then you want to compare between one of your sequence and every other sequence. So you need to have a library or framework to do that. It's a problem of BLAST, yeah. So BLAST sometimes is very difficult if your data is a big data, okay. So you need to be able to run it in parallel, and Biopython provide tools called BLAST, yeah, to deal with this problem, yeah, to deal with comparison between one sequence to another sequence. So in chapter 10, you will learn how to use BLAST capability inside the Biopython. So first you need to run BLAST on your query and get some output. And then second, parse the BLAST output in Python for further analysis. And what kind of analysis you can do. Of course, you can get into the content of this chapter, okay? So running BLAST over internet. So there's a function key BLAST in BioBLAST NCBI worldwide module. And also there is an online version. You can open the link here to see the online BLAST. It's kind of a CGI. CGI is a kind of web. So if we say CGI long time back, it's a kind of a web page with some logic behind. So this is a BLAST search tools. So means that from your sequence, you want to find nucleotide blast, protein blast, or you can compare like blast X and TB blast N, like translate nucleotide to protein or protein to nucleotide. Okay, very important search tools between protein and nucleotide. Okay, there is some other things also here like smart blast is like a predefined search or blast you can download and utilize. So this website, please take a remark on this website when you learn. And of course, there is also FTP from NCBI. Yeah. And here you can get the sample code of how to use BLAST in daily work. Okay. So from BioBlast, import NCBI worldwide, and then you can get the capability of BLAST. Yeah. So here the sample code, for example, from BioBlast, import NCBI worldwide, and then FASTA string, open mcode FASTA and read, and then result, you can call the key BLAST function to search or compare. Yeah, you can say also compare, or you can say, you can imagine search. Yeah, so based on my sequence, I want to compare or find out other capability, other sequence that is similar to my sequence. Okay, you can run BLAST locally, but I suggest you run it on your cloud environment, because once you deal with data from NCBI, you deal with big data, means that is huge data. During your computation locally, you may not be able to run any other things because your memory and your CPU is consumed by this process. Okay. So I will not cover code by code, line by line, because you are already in master degree. I just want to tell you that this is a very important library. What you need to understand, maybe this kind of diagram, that this is kind of a data class diagram for library, BLAST library. So from this diagram, you can sense what kind of features you can get from BLAST. Yeah. So take a look on this class diagram, so you can sense the capability of the library. So another tools also important other than BLAST is covered in chapter 11. So BLAST is the most popular. Okay. But other tools like FASTA, HMMER, and other things also complementary to BLAST. So read also this chapter. Yeah. Uh, like search IO capability here. You can also find out this chapter in Biopython chapter 8. So I think that's from me for this week. Continue to learn Biopython, because once you join the or you want to reproduce like SnakeMake or code-based library like SnakeMake and Nextflow, you will need this kind of tools. Also, if you're using Galaxy, sometimes this kind of library is wrapped inside the Galaxy. So Galaxy is a low-code drag and drop, but of course, it has some BLAST capability. So you will gain more understanding when using Galaxy if you know BLAST, okay, which is the basic is actually in Biopython library. Okay, so that's it. We will continue to learn Biopython after Lebaran holiday. I will hand over to Sinta and Fadin for reproducing pipeline session with Sinta and Fadin. Please.
Okay, thank you, Mr. Isman. Okay, good afternoon, everybody. Hello. Let's get started on practical session. Let me share the screen. Okay, previously we already learned about Galaxy and TeraBio platform, which is low-code platform. Does my screen already seen? Okay, let's follow this tutorial. Please open the link that I shared in chat column. Today we will learn about SnakeMake. This is platform that we can use using in bioinformatics, but it is not specific only for bioinformatics. It used for general cases also. And this framework is using code. Previously is low code, now it use code. Okay, please open the link that I already shared. And now our goal is learning the features that in snake okay, let's get started on basic example here. First,